So I thought that I would start my presentation today with a little bit of a quiz. And no need to have studied, and this is finals times for a lot of you in academia or with kids at home. So the question I have for you is, how many of you would are you gesturing at me? How many of you would describe yourselves as a quantitative kind of person, a number-driven person? And I need you to raise your hands because this sets up my whole talk, so work with me. All right, great, perfect. That's exactly what I expected, right? Because we're all scientists. We're driven by numbers, we're driven by data. We like to be able to correlate point A and point B and match things up that way. And so that's really where my mind went when I thought about how am I going to capture the year in review in HESI for the last 12 months. So I thought numbers. I thought, well, I could talk about the fact that we'd had close to 1,100 scientists from 250 different organizations participating in HESI programs in the last year. We could talk about the fact that we'd had 18 different workshops on 5 continents in the last 12 months alone. We could spend some time talking about the vibrancy of HESI's programs. So we have 15 committees, but we've launched about 14 or so new projects and programs within those scientific committees, again, just in the last year. I think a real testament to the microcosm of energy and creativity and productivity that HESI's existing science programs are. You know, we could talk about publication statistics because everybody likes those, right? We've had an average of 3 publications per month over the last year in the peer-reviewed literature. And some of those have had dozens of citations even just over the past year. Some of the others have been downloaded thousands of times, again, just in the last year alone. So these are really powerful statistics, right? These are powerful numbers. But they didn't feel complete. They don't really tell the whole story of who HESI is. They tell us how much we've done, to some extent what we've done, but they don't really describe why we're doing what we do and why it's important and how we've done it well. So what I would like to do is to try and convince you over the next 25 minutes or so that perhaps you all are not as quantitatively driven as you just raised your hands and told me that you were. And that is actually the reason why you're here at a HESI meeting in the first place. And furthermore, that is the reason, that same characteristic is the one that means that you are the kind of individuals that are truly going to have an impact on both the practice and the uptake of science that is going to impact human and environmental health going forward. So let me try and see if we can demonstrate that. And I'm gonna start with a story. And most of you know that I have 2 kids. I have a teenager, I'm not going to tell any teenager stories, and I have a 9-year-old who's in 3rd grade. And back in the fall, in a bit of a fit of working mommy guilt, I got roped into signing up for Career Day at school. And for those of you who don't know the Career Day concept, the idea is that a parent or a family member comes in and they talk about, this is my job, this is the training I've had, this is why I do or how I do what I do. So I get roped into this back in September when I was doing a lot of traveling. So it comes May and I'm getting emails saying come in for Career Day. So I do a lot of public speaking, I do a lot of working with crowds, but I was pretty nervous about being in front of a classroom full of 25 9-year-olds for 30 minutes times 3 separate classrooms, all by myself. So I was pretty nervous about this, but I had committed to doing this. So I show up and they bring us into the library, and just as a little aside, I'm thinking, who is my competition going to be? It's DC, it's going to be lobbyists and lawyers. It's not really that exciting for kids. But in walks this guy who is probably like 6 foot tall, covered in tattoos. He's a U.S. alcohol, tobacco, and firearms special operations agent. He's carrying, no joke, a battle shield, literally a battle shield that's like 2 feet wide by 3 feet tall, and a duffel bag that is filled with props that he's going to show the kids that could have like all come out of an X-Men movie. So this is what I am up against. But never fear, they had given me a really awesome little badge. So I had that going for me, and I had brought pictures of my cute dog, and that is my dog. So feeling well prepared, I soldier on into the classroom, and I spent some time talking to the kids about things that I thought they could relate to. So we talked about pets. We talked about chocolate. We talked about the toxicity of theobromine. So it ended up with discussions about exposure and discussions about dose and discussions about risk assessment. And the kids asked really, really great questions. They actually really engaged with the science. And I found the questions about the science of toxicology or the training that I had done to be pretty easy to answer. But then we got into discussions about what I do in my day job. And not being in a laboratory, it was a little bit harder for them to understand what science is out of that context. So I explained to them that at the HESI organization, we found that it's a really effective model to address important challenges. So if we want to make sure that the soap that you're using in the bathroom is safe to put on your hands, but also safe to wash down the stink for the fish in the environment. If we want to make sure that the cold medication that your mom gives you is also safe for you to take, as well as treating your congestion, those are things that we are tackling. And that we found a really effective way to do this is to bring together scientists from a variety of different technical disciplines from around the world, different areas of science training, and bring them together to try and tackle that problem. And that's a model we found has worked really, really well. So I'm feeling pretty good about this explanation, and little boy in the class, and we'll call him Alex, raises his hand, it's a true story, and he says, "So Mrs. Pettit, if this is a really good model, why wouldn't your company just hire all those scientists and put them in one building and make the magic happen that way?" I mean, think about it. It's a great question. I'm sure he wasn't thinking this way, but that's kind of what Bell Labs did in the 30s, or Los Alamos did in the 40s, or IBM did in the 80s, or Apple did in the 2000s. They picked sort of the best and the brightest from different areas of technical training. They put complex problems in front of them, and they let the magic happen. I'm not going to sit up here and tell you that that was not a good model, or that HESI is necessarily a better model. But I will argue, and I hope to convince you, that there's a reason that we don't operate through Alex's big, tall tower with just having everybody in the same uniform environment. And then that's really important to the way the organization works and why it is, in fact, that the numbers that we talked about at the beginning of the talk don't necessarily all add up. And in this case, that's actually a really great thing. So, to step back and explain why I think that is such a good thing for the organization, I want to reference a TED Talk that I heard a few weeks ago from a young woman named Amy Purdy. And Amy suffered from meningitis as a young teen, and she lost both of her knees, her legs below her knees, she lost her spleen, she lost her hearing, just tremendously devastating effects from the disease. And she has gone on to become, I think she's a snowboarder, she's a motivational speaker, she's just one of these really vibrant spirits. And when she talks about what it is that she's accomplished and how she's overcome these barriers, she summed it up this way and said that it's not about breaking down borders, but it's about pushing off of them and seeing what amazing places they might bring us. And I recognize that she was talking about a very different context than the HESI organization. But at the same time, I think if we look at this and we think about HESI, to me, I really see the power of HESI in this statement. To me, when we think about what it is that HESI brings to science, it's really not that difficult in the current connected environment to break down borders across different technical disciplines or to break down borders across different areas of professional employment. The opportunity here is to leverage those differences in context, to leverage those differences in insight, to leverage those differences in opinion and perspective, and to use them to generate science from a uniquely challenging environment. So how do we launch from that position of challenge? Not a place of homogeneity, but a position of challenge, and use that to generate science that is therefore uniquely positioned to address the really complex realities that we're all dealing with, and to deal with the complex realities of the user base and audiences that we're seeking to impact. That, to me, is the power of the HESI organization, and that is the opportunity that we have to move science forward. So what I'd like to do is to give you a few examples from this past year where we have demonstrated the ability of the organization to leverage those differences of insight and context to launch science really from that uniquely challenging position where we've got these conflicting sometimes parties or perspectives, and then to land the science in a way that it truly has a meaningful impact for the community that we're seeking to reach. So I'm going to give you a handful of examples, and I'd be happy to talk about all of our projects and programs, but I'm going to pick a few selected ones just from the past 12 months that really resonated with me personally, and you'll hear others during the presentations later today. So the first two examples are programs where HESI has actually brought forward some really important science that in this case is both very relevant to protecting pregnant women and particularly protecting their developing children, or in the case, or nursing mothers. So the first example, and some of you in the board heard reference to this earlier today, was a really exceptional workshop that the DART committee put on just a few weeks ago. And the point to make here is that the Food and Drug Administration in the US had launched some new labeling requirements for medications for pregnant and lactating women back in December. And those requirements changed the prior approach, which was a sort of an alphabetical labeling, tiered categories of risk, to a much more qualitative type of approach where you really needed to describe the preclinical data and give some pretty significant recommendations about how they should use that information to help guide the application to their patients. So this is a big step forward in terms of a change in practice. And what this committee is to be commended for doing was, in a period of less than 5 months from the time in which this new rule and draft guidance was released, they convened a workshop. But more importantly than just a workshop, they put together a series of case studies which allowed the participants in this meeting, both academics, regulators, government scientists, preclinical scientists, clinicians, to work with some mock data sets. So they got their hands down into the data. They looked at a variety of false preclinical data. There was some confounding clinical data that got thrown in as they worked through the case studies, and they became more and more complex. So this created a really unique environment for these scientists to get together, get their hands down into the information, and think at a very pragmatic level, how do we engage this information, and how are we going to use it to both protect and inform patients going forward? Second example, in terms of meeting emerging needs, again in the maternal and fetal health arena, relates to the largely highly growing area of biologics just in general. And this marketplace has more than doubled in the last 10 years globally in terms of new drugs. And certainly for many of us at HESI, it has particular significance recently in terms of making new treatments available for people that we care about. So one of the areas that we've also seen great development in the HESI organization with regard to biologics is the class of drugs that are these highly engineered molecules, some of which are bound to an Fc protein. And the value of doing this is that it enhances the half-life of the drug in the patient. So it gives them longer access to the medication. The challenge sometimes with this is that there is also an Fc receptor present at the paternal-fetal boundary. And that, under normal circumstances, allows for the transfer of beneficial proteins from the mother to the developing child. The potential risk here is you could have a circumstance where potentially the drug is therefore transferred from the mother to the child. The HESI DART committee is, again, to be commended for picking this up as an emerging need, recognizing that the science in this area is critically important in the patient marketplace, and I'm not talking about the financial, but I'm talking about the health needs that these kinds of drugs are serving is so expansive and growing so rapidly that there's a real need to continue to build the safety and biological database behind it. They have begun a first-of-its-kind program to develop and collect and generate information about the ontological expression of this Fc receptor in a variety of preclinical animal models as well as in the clinic. So this information, once it becomes publicly available, is truly going to inform the practice of developing these drugs in an increasingly safe way and help ensure that patients and clinicians and drug developers and regulators can make good and sound decisions about how these drugs are to be used in the audiences and their potential, those receiving them. Some other examples I'd like to give of the ways that HESI has helped to land the science in the last year relate to the development of solutions and tools. So beyond the idea of HESI just developing publications and having workshops, the HESI Risk 21 Committee, which is a program that for the last several years has been developing approaches and frameworks for accurate and efficient exposure-driven risk assessment, published a series of manuscripts within the last 12 months. But they recognized that this was necessary but not sufficient, and have since then developed a web tool that allows any user to get in and include both toxicity and exposure information, and then in a very visual way understand what the output is in terms of potential risk. They've taken it yet a step further, though, in terms of moving again from this position of challenge and really providing science in that, hitting that point where it has true ability to be taken up. So they, again, developed a series of case studies and trialed these at the Center for Food Safety at the FDA recently, along with EPA scientists. They'll be performing similar types of activities in China and Taiwan this coming fall. But again, these case studies provided an opportunity that would otherwise be simply unavailable for practitioners to get their hands into the data, to look at questions around assumptions, around application, around interpretation, around practice, in a way that moves us beyond the manuscript, moves us beyond a best practice document, and really allows for scientists to engage in a platform that would simply not be available were it not for the kinds of opportunities that HESI's created. Another example of a tool, not a web tool or a case study tool, but really a more biological tool, if you will, is one that has been developed through our immunotox program, or is in the process over the last year. So many of you are probably familiar with the T. Gennaro incident from about 10 years ago, where I think 6 or 7 first-in-human clinical trial volunteers almost died because of massive cytokine storm reaction in response to the drug that was completely unanticipated. The good news is that that kind of scenario hasn't happened again. The bad news, or the reality, is that there still is no real consensus on how you optimally screen for that. There's no standard assay, there's no core recommendation for best practices, and the HESI Immunotox Committee has gotten into the weeds around what are people doing, how are they doing it, and the bottom line is it's all over the place. But they, I think, have uniquely recognized that the opportunity here is not to suggest you must do it this way, because that's very hard to implement and very hard to make it happen. But what they have recognized, and again, this is one of these examples of moving from a really challenging environment with different perspectives, is that the opportunity here is to create some positive and negative biological control standards. So they have partnered with the National Institute of Biological Standards and Controls in the United Kingdom, and based on recommendations from the committee, they will be making available at the NIBSC some positive and negative control antibodies that can be used by anybody. Anybody can contact them and get access to these samples. So this doesn't mean everybody's going to run their assays the same way, but it does mean that at least there will be some point of consistency with which we can compare data, with which regulators can compare data, with which academicians and other scientists and clinicians can compare data. Again, providing an opportunity for solutions based upon the diversity of input that came from across these projects. Third category of examples that I want to highlight is to focus on the organization's ability over the last year to truly connect with global safety decision makers. So although we have only 15 projects and a committee budgets of total of about $2 million, pretty small, we have 4 separate programs that in the last year have been directly engaged with international decision-making bodies. So both the ICH as well as the IPCS through the OECD. So for example, on the OECD side, our bioaccumulation committee has been actively, with Michelle's leadership, been conducting a large-scale multi-site ring trial looking at hepatocyte in vitro metabolism. So this information is not only going to be valuable in terms of informing the way that we assess potential bioaccumulation of environmental chemicals, but it's also going to provide information that is going to reduce the burden of animals required in the future for testing for that very same endpoint that's intended to be environmentally protective. This is a program that's being done directly in support of an OECD test guideline. We also have a program through our Gene Tox Committee that is looking at the PIG-A mutation assay, for which the information is going to provide ultimately a more mechanistically relevant set of information about modes of genotoxicity, again in support of OECD. On the ICH side today, I don't know whether Brian's going to touch on it, but the cardiac committee, the cardiac committee is developing data that's being supported, being submitted and shared with the ICH as they consider new cardiovascular safety approaches. And similarly, we are supporting and providing information that's directly engaging in discussions on developmental and reproductive toxicity, use of second species, all those kinds of questions, again, directly linking to the ICH. So opportunities within a very small budget and a small organization to have a real impact by making sure that we're landing the science in the place where it has potential for uptake. The final one I want to touch on is this. That HESI over the last year, and Lori touched on it as well, has sought to build the science that we are addressing beyond that of simply that which comes from within the scientific committees. So we have launched a series of initiatives over the last 12 months through our Global Platforms Initiative, our Future Leaders Travel Fund, the SITE Initiative, that are seeking to build upon the knowledge and the expertise and the foresight that the organization has inherently in it with 25 years of collaborative experience, and to use those to build new partners, to build new relationships, and to help to share what it is that we have learned as an organization, but also to help bring new science into HESI as we go forward. So most of you who are here have been part of those really challenging discussions that take place in this HESI organization. You know that the science that we engage with is in many ways a lot more complex than that tower of science that my son's friend Alex was initially describing. But I think we have repeatedly demonstrated that the successes the organization has had are truly a function of the diversity of expertise and the diversity of experience that we bring to the challenges that we're addressing. We know that the opportunity to impact the ability to ensure that our children grow up healthy and strong, that our environments are clean and sustainable, that our friends and our family are living lives free from pain and disease, that these are complex and difficult problems. So I encourage you in that regard to continue to push each other, to continue to move forward, to distill down to those elements of the problem for which resolution can really yield an impact. So for now, I would ask that you forget about the numbers and instead focus on the difference. Focus on the difference that each of you can make as individual scientists, and focus on the difference that we all can make as HESI working together. Thank you.